Welcome everybody to this session, P15, from bench to bedside, preclinical research in IMI, ND, neurodegenerative projects. And my name's Craig Ritchie. I'm Professor of Psychiatry of Aging at the University of Edinburgh. And in a previous life was the academic lead of a IMI project called EFPIA, the European Prevention of Alzheimer's Dementia project. And towards the tail end of that project, IMI set up this coordinated action called Neuronet, recognizing that they had an incredibly powerful portfolio of studies or projects within the neurodegenerative space. And the existence of Neuronet was to help establish better connectivity between each of those projects so we could really not only learn from each other, but also share resources and knowledge between the projects. So our next presentation is from Jannis, who's based and joining us from Cambridge. And just as Jannis is bringing up his talk, I'll just introduce him to you. So Jannis got his PhD in Institute of Biophysics of Chinese Academy of Sciences, and during his PhD he determined the cryo-EM structures of light harvesting and reaction center core complex, an alternative complex 3 from Rosiflexus Kasten-Holtze, and developed an approach to ultra-structurally map subcellular localizations of proteins in 3D. In 2018, he joined Michael Durdert's group as a postdoc researcher and was also supervised by Jannis in the LMB. He focused again on cryo-EM structure determination of amyloid filaments from human brain and has solved many structures of tau filaments and alpha-synuclein filaments at high resolution. And I think critically important, I say this again as a clinical academic, identified the binding sites of a PET ligand on tau filament from Alzheimer's disease. So incredibly important, if you like, imaging and structural work, which I think has got great transformative potential or transformational potential. And also I noted was on the front cover of the front cover of Nature just a few weeks ago with some of his work. So congratulations, Jannis, for that. So I'll hand over to you and look forward to your presentation, structure-based classification of tauopathies and identification of PET ligand binding sites.
Okay, many thanks for the introduction. Hello everyone, my name is Jannis. It's a great honor for me to present here to show our recent work, which is about structure-based classification of tauopathies and identification of PET ligand binding sites. Accumulation of tau inclusions is the hallmark of multiple neurodegenerative disease. We call them tauopathies. Due to the alternatively splicing, there are 6 tau isoforms expressed in human tau, adult human brain, and 3 of them have 4 microtubule binding repeat. We call them 4 repeat tau, and 3 of them have only 3 microtubule binding repeat. We call them 3 repeat tau. And based on the isoform composition in the inclusion of certain tauopathies, they can be classified as 3R plus 4R tauopathies, 4R tauopathies, and 3R tauopathies. The Alzheimer's disease and progressive supranuclear palsy are the most common tauopathies. Actually, we have several ways to distinguish tauopathies. The main one, of course, would be based on the clinical features, such as dementia, personality disorders, and movement disorders. The development of position emission tomography imaging help us to allow us to see the tau inclusions in the human brain in centimeter resolution level when the patient is still alive. By using of postmortem neuropathology, people was allowed to see the antibody labeled tau inclusions in micrometer level. And recently, by using of cryo-electron microscopy, we could see the filamentous tau in atomic resolution And in this study, I would like to focus on how we use cryo-EM to solve the problem in neurodegenerative disease. Firstly, I would like to briefly introduce how we determine the first tau structures from Alzheimer's disease. And before doing this, I would like to thank all the patients and their families for the donation, because I think this is very, very important for us to understand our disease and all the other diseases. The tau filaments were extracted from the human brain, and then it will load to cryo-EM grids. And after the plant freezing, we could use electron microscopy to image the grid, and we usually took thousands of cryo-EM micrographs. And here is the representative of cryo-EM micrograph with full of Alzheimer's disease human tau filaments. And after a series of image processing, we could get a high resolution cryo-EM map. If we look at the cross-section of each filament, we could clearly see there are two types of tau filaments. One is parahelical filament, and the other one is a straight filament. However, the protofilaments from PHF and SF are identical, which is we call C-shape core. And the difference between PHF and SF is the inter-protofilament packing. During the past 4 years, not only for the Alzheimer's disease, my colleagues also looked at the tau filament structure in Pick's disease, chronic traumatic encephalopathy, and cortical basal degeneration. Surprisingly, although the tau filaments are made of the same tau protein, in each disease, the tau filament structure are different. And another thing is for the same disease in different patients, we always found the same tau filament fault. And this observation suggests that distinct conformers of filamentous tau define the different tauopathies. To have the overall idea of the relationship between the tau fault and the disease, we continue this cryo-EM work to other common tauopathies. The first one we were looking at was progressive supranuclear palsy, the PSP, which is characterized by both neuronal and glial tau pathologies. However, in the Alzheimer's disease, only neuronal tau pathology can be found. And the main symptom for the PSP is the movement disorders. We have checked 6 PSP cases and found there is a common tau filament structure, which we called PSP tau fault. Clinically, PSP is quite similar with cortical basal degeneration. However, in their structures, they are quite different. For example, the PSP fold into 3 layers, and the CBD fold into 4 layers. Therefore, it can be easily distinguished PSP and CBD based on the cryo-EM study. The globular glial tauopathy, the GGT, is another full repeat tauopathy. And there are 3 subtypes of GGT. We examined the 1 case of GGT type 1 and 1 case of GGT type 2 and found a common GGT fold. If we compare the PSP fold and GGT fold, we can see they are very similar because GGT also GGT tau filament also fold in 3 layers. The main difference between PSP and GGT is the C-terminal of GGT fold is pointing to the opposite direction and packing against with the end of repeat 4 in a hairpin-like structure. The atriophilic green disease, AGD, is another 4-repeat tau. The defining pathology is abundance of atriophilic greens. We have examined the tau filament structure from 2 AGD cases and found there is a common protofilament core. We call it AGT fold. AGD fold, the AGD tau filament also folds in 4 layers, which is quite similar to cortical basal degeneration fold. The main difference is the packing in N-terminal and C-terminals. Surprisingly, we also found AGD fold in other 3 tauopathies. The first one is aging-related tau astrogliopathy, RTAG, and the rest ones are the inherited version of tauopathies. The mutations are at +3 or +16 in the intron 10 of the tau gene. So therefore, the AGD tau fold is quite common in multiple tauopathies, 4 repeat tauopathies. And we also examine the tau filament structure from 2 inherited type of 3R+4R tauopathies, which is like Alzheimer disease. The disease called familiar British dementia and the familiar Danish dementia, FBD and FDD. Like Alzheimer's disease, FBD and FDD are all characterized by both intraneuronal tau pathologies and also extracellular pathologies. The difference is in the extracellular tau extracellular pathology is made of a different protein from amyloid beta in Alzheimer's disease, and the disease causative mutations are at the extracellular pathology, not in the tau gene. So we examined the tau filament structure from 1 case of FBD and 1 case of FDD, and found the tau fold are identical to those in Alzheimer's disease. They are parahelical filament and straight filament. The cryo-EM structure, which in this study together with the structure previously described, provide us the overarching perspective, and which suggests hierarchical classification of tauopathies based on their filament fold. The first layer of the classification is based on the isoform tau isoform composition. So the tau pathologies tauopathies can be classified as 3R+4R tauopathies, 3R tauopathies, and 4R tauopathies. At the second level, in the 3R+4R tauopathies, the CTE fold is different from Alzheimer fold. And although the Alzheimer's disease, FBD and FDD parts are pathologically different, they all have the Alzheimer fold. Therefore, we classify them as the same group. Also, at the second level of the classification, in the 4 repeat tauopathy, and there are 2 groups, the 3 layers group and 4 layers group. The CBD fold and HD fold are classified in the 4 layer group, and the PSP fold and GGT fold are in the 3 layer group. The structure-based classification not only help us to classify the tauopathies, which we already know, but also help us to identify new disease. Here is the example. We observe a different tau filament fold from a case from a patient who was diagnosed as PSP. Since these tau folds are similar to GGT and PSP tau fold, we call it GGT PSP tau fold or GPT. Histologically, these patients was subsequently found to be different from PSP because a lot of spherical full repeat tau and intraneuronal inclusions can be found in both limbic regions and other brain regions. Therefore, we call this disease the limbic predominant neuronal inclusion body for our tauopathies, the LNT. The second part in my talk is about tau position emission tomography imaging, which is used radioactive labeled small molecule to label the tau inclusions in the human brain when the people are still alive. This could allow us to do the diagnosis or and the longitudinal studies. Although many tauopathic ligand has been used in the patients, we still don't know how and where the ligands bind. Here, I would like to introduce a recent developed tauopathic ligand, APN-1607, which is also called PMPV3. It is a second generation tauopathic ligand. Based on the recent study in this ligand, we could see very limited off-target binding of this ligand can be found in health control, but a huge signal can be found in both cortical and subcortical regions in Alzheimer's disease. Therefore, this ligand is a promising tauopathic ligand. By using our cryo-EM, we managed to get high-resolution maps of the tau filaments from Alzheimer's disease with and without APN-1607. Therefore, we call them plus APN-1607 or minus APN-1607. We calculate the difference map. We calculate difference map by subtracting minus APN-1607 from plus APN-1607 or the other way around. So we got the difference map, and the orange signals indicate the signal from APN-1607. And in the paired helical filaments, the Alzheimer fold, we could see there are three ligand binding sites. The first one is in the arginine 349, and between the arginine 349 and glutamine 351, and we could find two binding sites between 351 and 353. Surprisingly, in the street filament, although they are they have the very similar C-shaped core, but not only we could see the exactly the same binding site to what we found in parent helical filament, we could also see actual densities, the binding site in the C-shaped cavity, which said that the different protofilament packing will have some effects on the ligand binding behaviors. And also, based on this binding site informations, this could allow us to develop better, more specific tau ligand in the future. So here comes the summary. By cryo-EM, we solve the structure of tau filament from PSP, GGT, AGD, R-tag, plus 16, FBD, and FTD brings. And this finding suggests over the hierarchical classification of tauopathies on the basis of their filament faults, which complements clinical diagnosis and neuropathology. We also identify a new tauopathy, INT, which with the filament features that are intermediate between those of GGT and PSP. And we also, the first cryo-EM study of PET ligand bound to amyloid filament showed the binding site of APN67 in tau PHFs and SFs. Knowledge of the binding modes may lead to the design of better tau ligands. So finally, I would like to thank people in the IMB, especially for Schultz Sherras and Michelle Goodrich. They both supervise these studies. And I would like to also thank other collaborators who provide us the brain tissues, the histology data, and PET ligand. And also I will thank the founder, the imprint and UKRI to fund this, to support this study. And also thanks for your attention.
So thank you so much, Yang Xu. That was a fascinating, fascinating presentation. And I'm sorry I missed your paper in Nature, but I'm going to go and look for it because I thought that your hierarchical structure of confirmation of different types of tau relative to different clinical disease states was absolutely mind-blowing. I think, you know, we do tend to, as you know, describe a lot of the diseases through their clinical phenotype. And I think searching for a biological basis that distinguishes them at a very, and presumably at a very early stage in the course of the disease, this, you know, conformational changes in the tau protein will drive downstream pathologies that lead to the clinical phenotype. So I thought that was absolutely fascinating. One thing I was, you know, I'm a huge fan and advocate for PET tau. I think that's going to be game changing as we use it both clinically and for research. One of the things I'm trying to get my head around though is that your APN 1607 ligand, in some ways, and correct me if I'm being a bit stupid here, but you'd almost need to probe for the particular tau structure with a particular ligand if you knew what that tau structure was. And I guess clinically it's saying like, is it PSP? Yes, no. So we find a ligand that will, you know, find that confirmation, rather than being able to give a ligand which will help with the differential diagnosis, if you like. Does that make sense? If you know what you're looking for, you can go and find it. But if you don't know what it is, a specific tau ligand isn't going to really help you. Does that make any sense?
Yes, it's true. It's true. Yes, because based on the recent paper, for this particular ligand, APN627, it has proved that it can bind to Alzheimer's disease and also PSP. So therefore, we subsequently do the cryo-EM study of the Alzheimer's disease for this ligand. So we are yeah. Before we do the cryo-EM study, we must know the clinical.
Yeah.
That is the case. Yes.
Because I guess if you know we've got a keen interest in chronic traumatic encephalopathy. We work with contact sports athletes who've retired, and I guess one of the things is, you know, if you give a PET tau ligand to somebody with CTE with APN 1607, would you expect it to be a inverted commas negative scan?
This could be. I think firstly, we didn't do any cryo-EM with the CTE filament with this particular PET ligand. So we don't really know how the binding behavior for this PET ligand. And another thing is that For the CTE fold and Alzheimer fold are quite similar, and in the same binding site, we could see the side chain groove for the binding site in the Alzheimer fold, which will be very narrow, which will be narrower in CTE fold, which might explain we could see a negative scan for this particular ligand in CTE patients. I think this is reasonable to have this.
So I've just got overexcited and I forgot to look at the time. So we're running out of time, I'm afraid. But I'm going to look forward to watching your work and career as a consequence develop. I think it's a fascinating area and it's going to open up so many avenues for us, both research, clinically, and otherwise. So thank you so much and good luck. So I think just as we're transferring over to Jannis's presentation, which is the last of our presentations today, I'll just introduce Jannis Wißfeld. I hope I've pronounced that properly. Jannis works in Bonn. He's working on the PHAGO project. He's a bachelor degree in molecular biology at the University of Mainz, a master's degree in life and medical sciences at the University of Bonn, and a PhD degree in biology at the lab of Harald Neumann University of Bonn, in the University of Bonn, and worked on how variants of CD33 influence microglial activation. In his presentation, Alzheimer's disease protective variant of CD33 increases microglial activation. Jannis.
Yes, thank you very much for the nice introduction and thanks for having me today and giving me the opportunity to present my data here. So today I will shortly talk about the PHAGO consortium and here also in particular what they do, a little bit on Alzheimer's disease and then on my work in the PHAGO consortium, which is creation of some model systems and also then some results of those model systems and will sum up that shortly. So the PHAGO consortium consists as many of those consortia of some EFPIA partners, SMEs and universities, academia. We are around the world actually, with many in Europe, some in the US. And our goal here is to identify the roles of TREM2 and CD33 in Alzheimer's disease. Here, first of all, to provide some tools or reporter cell lines or iPS-derived microglia cell lines to study those two receptors in Alzheimer's disease. So on cellular and molecular level, Alzheimer's disease is characterized by these characteristic A-beta plaques, neurofibrillary tangles in neurons, and reactive microglia. And in the recent years, actually, the research focus switched a little bit from the A-beta and tau to these reactive microglia as main contributors of the disease. And these microglia can act in two different roles. It's actually a little bit more difficult here, but for reasons of simplicity, I just show you two phenotypes, which is one, the anti-inflammatory microglia phenotype and the pro-inflammatory phenotype of the microglia. These are considered to be neurodegenerative, whereas the anti-inflammatory are considered to be neuroprotective. But in reality, we have a fluid transition, and that depends highly on the stimulus they encounter. One of those signaling axes shaping the microglial phenotype is the ITIM and ITAM signaling axis. So we do have here members of the ITIM signaling receptors, namely TREM2, which then become activated and lead to cellular activation here by cytokine secretion, phagocytosis, and oxidative burst. On the other hand, we have these ITIN signaling receptors, to which cyclins belong, and CD33 is one of those cyclics. And these kind of modulate the response or the microglial activation then by recruiting of tyrosine phosphatases and dephosphorylation of those pathways, and thereby modulating microglial activation. So CD33 was recently identified to be contributing to Alzheimer's disease. And here in particular, there was a protective variant of CD33, which showed increased phagocytosis, or those microglia showed increased phagocytosis. So we were interested in studying CD33. And to do this, we need a let's say, reporter cell line in order to find high affinity ligands to be able to modulate the microglial response in Alzheimer's disease. And this is what we did here. We generated a CD33 reporter cell line system. And we did that within not very common way. We used that intracellular domain of tauopathy, which is the activatory protein or the activatory pathway protein, instead of the intracellular signaling motive of endogenous CD33, which is an inhibitory motive. And this we did because it is easier to monitor. And the goal was here to create a reporter cell line which can be used for ligand screening approaches. So this reporter cell line we did in two different variants. One is the full-length variant here with a big M, and the other one is a delta E2 variant. This is the Alzheimer's disease protective variant. And the reporter cell line works as follows. So when we activate it with either an antibody or a ligand of CD33, we get that a recruitment of the kinase SYK to tauopathy here, which then in the end leads to intracellular calcium increase. And this can be easily measured, and it's a quite common tool in industry to identify ligands for receptors. We also transfected genetically engineered GMP, which fluoresces upon calcium binding. So using that tool, we were able to identify out of a panel of CD33 binding antibodies, two antibodies, here are clone 1C71 and P67.6, which were able to activate the CD33 full-length reporter cell line next to the positive control ATP here. But the Alzheimer's disease protective CD33 variant was not activated by those antibodies at all. So because our reporter cell line is a quite artificial system and we aim actually to modulate microglial responses, we of course were interested whether these antibodies now are also suitable to modulate these responses in a more suitable model system. And for that, we used iPSC-derived microglia, so a second model we generated in Fargo. And these are produced by stem cell culture and embryonic body formation. And then we mixed them with a little cytokine mix, which they have some, plate them then in plates. And after a certain time, which is 4 to 6 weeks, they give rise to microglial cells, which can be easily harvested and seeded. And these we used them for the experiments and looked at the phosphorylation of ZYK. So now this is the opposite pathway, right? CD33 is modulating this pathway. So if we activate CD33 in the endogenous model, in this iPSC-derived microglia, we do or should see a decrease in phosphorylation of ZYK. We did that by co-treatment with an activatory antibody against TREM2 and an active or the putative activatory antibodies against CD33. As you can see in the wild type, which is the full-length CD33 expressing cells, we do see a decrease in phosphorylation of ZYK mediated by antibody 1C71 and P67.6. And this was not true in a CD33 knockout line or the Alzheimer's disease protective variant CD33 delta 2, showing us that our reporter cell line is working quite nicely and might be used for ligand screening approaches. So to sum this up, we have identified two antibodies activating our reporter cell line system and two who does not activate it and could confirm that in the iPSC right now. So the second part is about the role of CD33 in the human microglia. So here we use these iPSC-derived microglia model to study CD33 and use then either the wild type line, the knockout line, or the CD33 delta 2 line. And then we were interested in what is happening downstream of CD33. As I said, again, we used our quite nice model system here, the iPSC-derived microglia. And the first we investigated was phosphorylation of ZYK again, so as a direct partner downstream. And we could see that here in gray, the wild type, the knockout in red, and in blue, the CD33 delta 2, so the Alzheimer's disease protective variant. We see that if we knock out CD33 or express that Alzheimer's disease protective variant, we do see an increase in baseline phosphorylation of the here. We was also interested, of course, in how the system works if we trigger it. So we use an activating antibody for TREM2. And as you can see here, so the activating antibody for TREM2, it increases the phosphorylation. But when the CD33 knockout, we have a much higher increase. And this is also not true for the delta 2 line. And the same is true if we use an antibody against Fc gamma receptor, showing us in the end that we have an exacerbated response in the knockout, but this is not true for the delta 2 variant. Looking at phagocytosis data here with some labeled bacterial particles, we do see an increase in phagocytosis in the knockout and the delta 2 line. This is already known from literature. And the same is true if we're looking at disease-specific stimulus A-beta here. So we use aggregated A-beta, feed it to the microglia, and we do see higher increase in phagocytosis in the knockout and the delta 2. And the last parameter I looked at was an oxidative burst, so the radical production which occurs upon phagocytosis. And using the bioparticles, we can increase that in the microglia, and using a scavenger, we decrease it again. And as you can nicely see, the knockout produces much more radicals upon encountering or phagocytosis than the wild type or the delta 2. So summing this part up, so the interesting part is here, CD33, the full-length form decreases inflammation, while the Alzheimer's disease protective variant increases cellular activation and phagocytosis. But it's not a knockout, so we do see difference between a full knockout and the delta 2 variant. So with that, I'd like to end my talk today and thank, of course, all our collaboration partners from Fargo, the neurodegeneration group from Bonn, and you for your attention.
So thank you so much, Jannis, for that presentation. I must admit, I always struggle with microglia and TREM2 and CD33. I'm a clinician. I'm not a lab person, but I do wonder sometimes about the complexity of the interplay between all of these different elements and the cell lines, what the potential is for therapeutic intervention. Are you suggesting that CD33 upregulation may be a good target to treat or prevent Alzheimer's disease?
So in case of CD33, it's quite complex because we do see for the protective variant, we do see an increase in microglia activation. So apparently at some point of the disease, microglia activation is beneficial. Yes. And at some point it overturns and gets detrimental. And this is our current hypothesis. So you have in early years where the disease develops, you don't have a clinical phenotype, you don't have any symptoms. There you need that microglia activation to clear off A-beta plaques and keep the brain in homeostatic state. And at some point, when that fails, you actually then get a reactive microglia phenotype, which then in the end leads to the symptoms and Alzheimer's disease.
Yeah,
so,
I think, yeah. And I think, sorry, the definition of, in a patient at some point is going to be one of our biggest challenges.
That is true. That is true.
Yeah, and do you think there's a potential for because I can't imagine again, to use your term, at some point, and I agree that's the best term. Do you think that's something which can be picked up through a biomarker? I can't imagine a clinical phenotype is going to help us, or do you think it'll be something else, or a combination of different elements?
Yeah, this is a really good question, because I think that is one of the most challenging things we are up to right now, so to find the right time point to intervene, actually, right? So we don't have an early onset biomarker. We do have SPF tau, or tau or A-beta, sorry. So that gets less because it builds up in the brain. But the question is whether that's already too late, right? If it's building up the plaques in the brain, it gets less in the cerebrospinal fluid. So I don't know whether this is the right time point to intervene still, or whether it's too late. But yeah, so we definitely need some better biomarkers for early intervention.
And I think one of the things is that, sorry, I'm being told the room's going to automatically close in a second. So I should probably, we'll take this offline because we work very closely with Fago from the EPAD project. But I think it probably just before we get shut down, I'll just thank you again, Jannis, for that wonderful presentation. I found it particularly fascinating, and I think these talks will be available online for a period of time for those who weren't able to make it into the session today, because it was a parallel session. Also a huge thanks, obviously, to Alzheimer Europe, to Chris and Sophie for supporting the session today, and Neuronet for the great work they do in trying to bring all of these IMI projects together. So thanks again to all the 4 presentations. Absolutely brilliant. Thank you.